Good afternoon. I'm Dr. Greg Eastwood. I'm the director of the Inamori International Center for Ethics and Excellence. We'll call it the Inamori Center for short. I'm pleased to welcome you to the lecture and the symposium and conversation to follow the lecture that's in association with the first Inamori Ethics Prize. I'd like to also recognize groups of people that are in the audience. We have students from area high schools, including the John Hay High School. The John Hay High School students came to visit us yesterday for a special session. Would the John Hay students just wave your hands so we can see you? There you are. Special welcome. Are there other high school students with us today? If you can wait. Oh, good. I'm glad you're here. And of course, we have members of the, yes, let's not. We have members of the Case family, students and staff and faculty. We have people from the community, from other colleges and universities, and we even have some people from overseas, mostly Japan. So I'd like to recognize several members of the audience today. First is Dr. Kazuo Inamori, president of the Inamori Foundation. He's accompanied by several other people from the Inamori Foundation, Dr. Shinji Fukukawa, Vice President of the Inamori Foundation and former Vice Minister of the Ministry of International Trade and Industry. Dr. Heisuke Hironaka, Director of the Inamori Foundation and Professor Emeritus of Kyoto University. Mr. Toyomi Inamori, Senior Managing Director of the Inamori Foundation. Mr. Kazumasu Umemura, Executive Officer of the Kyocera Corporation. Mr. Mikio Okuno, Senior Advisor for the International Department of the Inamori Foundation. Mr. Takanori Kutsuna, Secretary General of the Inamori Foundation. And Mr. Kiyohiko Kogoshi, Director and Manager of the International Department. Also, please recognize Dr. Francis Collins, recipient of the Inamori Ethics Prize, and his wife, Diane Baker. Diane will be joining us a little later. Yes. Barbara Snyder, President of Case Western Reserve University. Mr. Frank Linsalata, Chair of the Board of Trustees of the University, and his wife, Jocelyn. And Mr. Charles Koch, Chair-elect of the Board of Trustees, and his wife, Katie. A number of individuals and organizations have helped to make this afternoon possible. We're grateful to the Cleveland Foundation, to the St. Luke's Foundation, the Mandel Foundation, Ewing Cole Architects, Mr. Alan H. Ford, Dr. Theodore Castell, and Richard and Judith Watson, all of whom combined to support the lecture and symposium that you're engaged in now. Let's thank them. I also especially want to thank Mr. Tom Zenti, who's the CEO of University Hospital. He is not here right now, and he is represented by Dr. Achilles Demetrio, but the University Hospitals of Cleveland have been very generous, and they are the overall title sponsors for all the events today and this evening. Let's thank Mr. Zeni, University Hospital, and if Dr. Demetrio is here, please stand up. When you have the opportunity, I encourage you to thank all of our sponsors when you see them personally. Now I'd like to tell you about the Inamori International Center for Ethics and Excellence. The purpose of the Inamori Center is to foster ethical leadership around the world. Thus, although the center has a global purview, we believe that the world begins right here, in this room, in this building, on this campus, in this city, and extends outward. We engage in research and scholarship, and we sponsor symposia, lectures, other means of ethical discourse. We are creating internationally recognized programs devoted to ethical leadership and the development of future leaders who, in the words of Dr. Inamori, will serve humankind through ethical deeds rather than actions based on self-interest and selfish desires. The Inamori International Center for Ethics and Excellence began operations about 2 years ago as the result of a generous gift from Kazuo Inamori, president of the Inamori Foundation, whom you just met, and founder of the Kyocera Corporation and KDDI Corporation. Dr. Inamori believes that people have no higher calling than to serve the greater good of humankind and society. Case Western Reserve University has had a long and productive relationship with Dr. Inamori and the Kyocera Corporation, and it all began with a remarkable person, one of our most distinguished members in the School of Engineering, Dr. Arthur Hoyer. In 1985, Dr. Hoyer became the Kyocera Professor of Ceramics, one of only 3 such professorships in the country. Professor Hoyer also is a member of the National Academy of Engineering. Since the 1980s, Dr. Inamori has visited our campus several times. In 1995, he gave the T. Keith Glennon Lecture titled My Leadership Paradigm, and the text of that has helped guide me as I have led the Inamori Center. We think that Dr. Inamori saw at Case Western Reserve University a respect for ethics as a value which lies behind the programs of the various schools, and that a center which fosters ethics, leadership, and internationalization would improve on the strengths that already exist here. The mutual interests of Dr. Inamori and this university in developing such a center culminated in an agreement to create the Inamori International Center for Ethics and Excellence. Here we see Professor Hoyer and Dr. Inamori enjoying the moment at the time of the signing of the agreement to create the Inamori Center. In May 2006, Dr. Inamori attended the university commencement and was granted an honorary degree, Doctor of Engineering, by Case Western Reserve University. And on May 7th, 2007, the Inamori Center was dedicated. Here are President Snyder, Board of Trustees Chair Franklin Salata, Dr. Inamori, and me cutting the ribbon. Notice we're all surgically adept. One of the ways in which the center fulfills its purpose of fostering ethical leadership is by awarding a high-profile ethics prize, and that is why we are convened for these events today. The Inamori Ethics Prize honors an outstanding international leader who has demonstrated exemplary ethical leadership and whose actions and influence have created something that will improve the condition of humankind. Now I'd like to tell you a little bit about our Inamori Ethics Prize recipient. Francis S. Collins has led the Human Genome Project for 15 years. During that time, Dr. Collins has paid particular attention to ethical issues related to the Human Genome Project. Also, the Human Genome Project holds enormous potential for the improvement of humankind. Thus, Dr. Collins fits perfectly the criteria for the Inamori Ethics Prize. Of course, Francis Collins has not always been a physician-scientist. Here we see him at age 2 in 1952, about to respond to a reporter who asked, "Do you think that Watson and Crick have a chance to win the race to determine the structure of DNA?" Francis is reported to have said something that sounded like yes, and sure enough, Watson and Crick did announce the structure of DNA the following year in 1953. Francis Collins was raised on a farm near Stanton, Virginia, the youngest of 4 boys. Listen to what he says about that in his book, the language of God. I was raised on a dirt farm in the Shenandoah Valley of Virginia. The farm had no running water and few other physical amenities. Yet these things were more than compensated for by the stimulating mix of experiences and opportunities that were available to me in the remarkable culture of ideas created by my parents. He dedicates the book to his parents, who he says taught me to love learning. Here we see Francis at age 6, contemplating his next stimulating experience or opportunity. Looking back, Dr. Collins says, I was born into this happy mix of pastoral beauty, hard farm work, summer theater and music, and thrived on it. At first, he was homeschooled by his mother, whom he regarded as a remarkably talented teacher. He says, learning was never something you did because you had to, it was something you did because you loved it. At age 10, Francis moved into town and entered public schools. He says, at 14, my eyes were opened to the wonderfully exciting and powerful methods of science. Inspired by a charismatic chemistry teacher, I discovered for the first time the intense satisfaction of the ordered nature of the universe. The fact that all matter was constructed of atoms and molecules that followed mathematical principles was an unexpected revelation, and the ability to use the tools of science to discover new things about nature struck me at once as something of which I wanted to be a part. Here's Francis at age 16 in his senior photo at Robert E. Lee High School, Stanton, Virginia. He seems to be gazing beyond the photographer to the glorious future that awaits him. But Francis Collins is multi-talented and adept at working into his life many interests, intellectual and otherwise. Here we see him playing his guitar in a role in his mother's play, The Lady and the Unicorn. In 1966, Francis entered the University of Virginia, where he not only played in a band, but also earned his Bachelor of Science degree in chemistry. From there, he went to Yale University and earned a master's and doctorate in physical chemistry. Next, he entered the University of North Carolina School of Medicine and earned his MD. After that, he engaged in several years of training as a resident in medicine, followed by a fellowship in human genetics and pediatrics back at Yale University. After all that preparation, Dr. Collins arrived at the University of Michigan in 1984, where he rose to become professor of internal medicine and human genetics. Here we see him in 1990, running a meeting in his laboratory at the University of Michigan. Now, I bet you thought that was a script for the movie Gattaca on the blackboard behind. But no, it is a script, but it is for a portion of the gene that results in cystic fibrosis, one of the several genes that Dr. Collins and his colleagues have discovered. I would add that the dean of our own School of Medicine, Dr. Pam Davis, uses that information in her research on the genetic treatment of patients with cystic fibrosis. Early in his career, Dr. Collins volunteered as a physician in a missionary hospital in Nigeria. That experience influenced him to advocate for applying the results of genomic research to improving the health of people everywhere, not just in developed countries. Perhaps we'll hear more about that later this afternoon. In 1993, Dr. Collins became the director of what now is known as the National Human Genome Research Institute, and of course the director of the Human Genome Project. That remarkable international project culminated in April 2003, just 10 years, with the completion of a finished sequence of the Human DNA Instruction Book containing over 20,000 genes. Along the way, Dr. Collins has been an active opponent of the misuse of genetic information. Here we see him testifying before Congress in support of the Genetic Information Non-Discrimination Act, which protects people from discrimination by health insurance and insurers and employers on the basis of genetic information, and which President Bush signed into law last May. Francis Collins has received hundreds of honors, including a string of honorary degrees, but the honor that has to be special for him is his receipt of the Presidential Medal of Freedom, the nation's highest civilian award. Here is President Bush bestowing the Presidential Medal of Freedom on Dr. Collins in the White House last November. The inaugural recipient of the Inamori Ethics Prize is a remarkable multidimensional person, a physician, a scientist, a husband, a father, a musician, and a Harley biker. Before we hear from Dr. Collins, I have asked Dr. Joseph Nadeau, chair of the Department of Genetics in our School of Medicine and a longtime friend and colleague of Dr. Collins, to put some of Dr. Collins' achievements in the practical context of science and medicine. I refer you to the middle of your program where Dr. Nadeau's biography appears.
It's a pleasure to share a few thoughts and words about Francis. I first met him years ago in the early days of the Human Genome Project when conferences, when scientists got together to both talk about the research that was ongoing and what the possibilities were for the future. At these conferences, pioneers in medical genetics, molecular biology, technology development, some of whom were Nobel laureates, often filling the front rows, schemed together to build this new discipline that we call genomics. And in this setting, there was a tall young scientist that appeared every year, that showed up every year to talk about his research, and in particular about the pioneering work that he was doing to discover disease genes, like the cystic fibrosis gene, that some of the researchers here at Case, not only Dean Davis, but Mitch Drum and others have worked on. But even in those early days, many of us recognized that not only was Francis an exceptional researcher, expert in the technical details of doing the complex work of genomics, which involves both lab work and bioinformatics, he also understood the incredible opportunities that genomics meant for human health, but also the risks for harm if this information was not used appropriately. This unique combination of scientific expertise, vision of the future, and obvious leadership abilities led to a very fast track in academia to his position as director of the National Human Genome Research Institute. I had the privilege of serving on Francis's advisory council in the heyday of genomics when the work that he was overseeing transitioned from pilot studies of feasibility. What could we do? What might we do if technologies worked, if the funding were available, to full-scale sequencing of the human genome. It was perhaps one of the most exciting times in all of the history of science. But from this platform of leadership at NHGRI and as an international leader in the field, he put together combinations of academic and business leaders and pharmaceutical companies to coordinate their efforts to sequence the human genome, the genome of other species, so that we would know who we are genetically, we would understand better our genetic and biological relationships with all other species on the Earth, and we would have for the first time insights into disease processes that we could only guess at a few years ago. From this position, among the things that strike me most is that he led to an international effort to make sure that the sequencing information was made available literally as it came off the sequencing machines, that it was free access. Everyone had access to this information because he understood that this information, even in its fragmentary forms and as it built up until we finally had the complete sequence of the human genome and other genomes, that information stream was fundamentally important and would transform our understanding of human biology and of disease processes, and it had to be free and it had to be immediate. He also fought to protect the use, to guide the use of genetic information, and to make sure that the information about us we had control over, and that we had appropriate relationships then with government, with industry, with insurance companies, with employers, about who had access to the information and what was appropriate use of it. Third, he recognized very, very early on the important social, ethical, and legal issues that were implicit in gathering this information about not only the human genome, but about all of our genetics. And so he built research programs and policy for him to make sure that these issues were very prominent, that we were talking about them, thinking about these issues in real time. because he knew that the use of this information for good was also very tricky, and it was important that we pioneer this at the same time that we do the good science, and it was important that we get it right. Finally, he understood that this information would have profound implications about how we understand our relationships among each other. We understand now better the genetic variation that we all see among all of us, and also the deep cultural differences that make us distinctive and interesting. But at the end, all of that genetic information reveals, too, how we're all part of one human family, how profoundly similar we are to each other. And that, to me, is very transformative, because in the past, our misunderstanding about our relationship has been used, sometimes in good ways, but often in harmful ways. Finally, Francis belongs to a long legacy of outstanding scientists who readily reconcile the fundamental tenets of science, namely rationalism and empiricism, with a deep religious belief. That was alluded to earlier in some of Greg's comments. And no doubt in the next stages of his career, Francis will continue leading us in important and good directions. So it's been a pleasure knowing Francis over these many years, and it's an honor to introduce him. Francis.
Thank you, Joe, for a very kind introduction, and thank you, Greg, for that amazing tour through my 58 years. I had no idea how much of it would be on display, but it was very graciously done, and I appreciate all of the effort that went into preparing for this really grand symposium on a grand day. And most of all, I would like to say thanks to Professor Inamori and to his wonderful team from the Inamori Foundation for having established this prize and giving me the great honor of being the first recipient. In brief conversations with Professor Inamori at lunch today, I learned of the way in which he has valued ethics in his own career and therefore sought to establish this center and this prize as a way of spreading that wisdom here at Case and around the world. And I think that's a wonderful testimony to a truly philanthropic person. So thank you very much to the foundation, and particularly to you, Professor Inamori, for this wonderful honor. In thinking of what to say on this occasion, I thought it would be appropriate to talk about what the science of the human genome is telling us about our own instruction book, but to focus particularly on some of the ethical challenges that this has put in front of us, challenges that are in the process of being addressed, but I don't think any of us would say that we're all done with this, because the science keeps changing, and so the ethical challenges as well keep evolving and continually need attention. So I'll go through some of the science, and then I'll go through a list of what I would consider some of the more pressing ethical questions and give you a bit of a snapshot of what's happened with those, but obviously make it clear that there's lots more work to be done here. I'm delighted there are many students here this afternoon, whether you're in the laboratory doing recombinant DNA experiments, or whether you're thinking about those consequences from the perspective of philosophy, ethics, social science, and law. We need all of you in this next phase, because the exponential increase of knowledge is not going to stop anytime soon. And that means we need an exponential increase in participation. So I'm glad you're all here today to be part of this conversation, and I hope we can have a really nice dialogue as the afternoon goes on. If I actually get around to finishing what I want to say, we'll have time for that. I should have also said what a treat it is for me to be here this afternoon with some of the people that I've had the privilege of training here in the audience, who are now on the faculty here at Case, and that includes my first graduate student who became my first postdoctoral fellow, Dr. Mitchell Drum. It includes Dr. Anna Mitchell, also a very talented physician-scientist, and it includes Dr. Peter Scaccieri, who arrived here only a couple of years ago. All three of these individuals doing really cutting-edge research in human genetics, and I'm delighted to see the way in which this town and this university and this academic environment has embraced them and given them a chance to do great things. So that's particularly nice for me to come to this little outpost of my own lab. There's no greater triumph or privilege or happiness that a professor in a research environment has than to see their kids grow up and achieve great things. So this is really a wonderful thing for me. It's also wonderful to have here with me this afternoon my niece, Natalie Podner, and her daughter, Rachel, and my nephew, Mark Collins. Both of them live here in Cleveland and were gracious enough to do something unusual. I'm sure they had other things they were supposed to do today, and it's wonderful, Mark and Natalie and Rachel, to have you all here with us as well. And somewhere out there, I think, is my wife, Diane, to whom I should say thank you for the way in which she's supported all of the efforts that have gone. I see her, she's right there. All the efforts that have gone into the Human Genome Project and its leader for the last 15 years, keeping me sane and keeping me in a place of wise advice, which she, as a trained genetic counselor, has often provided when I wasn't quite sure what the right answers were. So thank you, Diane. Well, the title of this, The Promise and Peril of the Genomic Revolution, sounds pretty dramatic, but we are living in dramatic times. And maybe revolution is a strong word, but I think it's fair to say that what's happening right now with our revelations about human biology based upon reading our own instruction book are unparalleled and have the promise of transforming medicine in a way that hasn't happened perhaps since the introduction of antibiotics. It is not happening overnight. It has already affected quite a lot of people who live in particular circumstances where studies of the human genome have had a direct impact upon their medical care. But for many of us, that moment of the genome invading your own potential medical future hasn't quite happened, but it's coming pretty quickly. And I want to explain why I say that with confidence and where I see both the promise and the peril residing. As you heard, we went from having the structure of DNA deduced in April 1953 to almost exactly 50 years later, the completion of the goals of the Human Genome Project, April of 2003, laying out those 3 billion letters of our own DNA code in a completely accessible fashion. And of course, that had been the case all along. It's not like we waited until 2003 to dump it all into the computer databases. It had been accruing ever since 1996. And I want to stop right now and say that while I'm standing here telling you about that, my job here was to be rather like the conductor of the orchestra. And all of these were the people who were playing the instruments, and beautiful music they were making indeed. This is a photograph of some of the leaders of the 20 genome centers in 6 countries that successfully achieved those goals of the Human Genome Project ahead of schedule and under budget, believe it or not, in 2003, including, I might say, because of the connection here today with the Inamori Foundation, including a substantial contribution from our friends and colleagues in Japan, as well as those in China and the United Kingdom, in France, and in Germany. The U.S. had the privilege of taking the lead in this enterprise, but it was very much an international project, and my job was to be like the field marshal or the project manager or the cat herder or whatever you want to call it, to try to be sure that we had shared goals that were well articulated, that we stood up for immediate data release, that we didn't hoard any information that might help anybody even for 24 hours, and that we maintained data quality standards of the highest possible level so that if you found our data on the internet, you could trust it to be right. And all of these folks made that happen and didn't worry too much about who got the credit because they believed in the promise of this project to transform our understanding of human biology. And they were right to do so. So I guess in that regard, I'm fond of this quotation from Woodrow Wilson because it very much seemed like that's the way things were for my 15 years with the Genome Institute, using whatever brains I had, but doing a lot of borrowing from all of these other gracious people who gave freely of their ideas, their intelligence, and their hard work. Now that was great because that gave us a foundation of the part of the genome that we all share, which is more than 99% of it. But what about the variable part, and how could we use an approach to that to try to understand the causes of illness? After all, virtually all diseases have some hereditary contribution. Some of them, like the ones in the pink curve here, are inherited in a fashion where a single gene gone awry is capable of causing rather predictably a clinical problem, cystic fibrosis. fibrosis, Huntington's disease, sickle cell anemia. Those are conditions where one gene leads to a disease if it is misspelled in a certain way. And the Human Genome Project provided the kinds of tools to make it possible to find those genes at a prodigious rate, as you can see from this curve. The scale here on the left in pink tells you how many different human Mendelian or single gene diseases were figured out. That is, the genes were identified. And you can see between 1990 and 2000, that jumped up to almost 2,000. It's now well over 2,000 that have had this kind of observation made about what gene is involved. But look, so we could smile about that, I guess. But look at the curve in the bottom there with the filled in blue triangles. Those are human complex traits. What's a human complex trait? Well, it's a condition which is not inherited in a simple fashion that Mendel would approve of, but it is inherited in the sense that it runs in families. That would be diabetes. That would be Alzheimer's disease. That would be heart disease. That would be the common cancers. And now the scale is the one on the right, and these authors in 2002 basically only believed 7 success stories in that regard, just 7 out of many, many years of hard work on the part of many people. So we couldn't smile about that one. Why was that so hard? Well, it's because you're looking, in a case of, say, diabetes, not for a gene. You're looking for many different genes, each one of which may harbor a genetic variant that increases your risk by a little bit, but it's certainly not deterministic of whether you're going to get diabetes or not. And it's only a combination of genetic risk factors and the environment, in diabetes, of course, that being diet and exercise, that determines whether you're going to slip over that threshold and become diabetic or whether you won't. And that makes this orders of magnitude more difficult to unravel than it would be for something like cystic fibrosis, where you're looking for a single gene. So why were we so unsuccessful? Well, basically, until about the time this diagram was made, the way we went after genes involved in common complex traits like diabetes was to try to guess. We'd say, well, you know, diabetes, okay, that's insulin, that's glucose. What are all the genes we know that are involved in insulin and glucose metabolism? Let's check them out one by one and see if we can find variations that seem to predict who got it and who didn't. And it was devastatingly disappointing. And the problem really was we were doing what's called a candidate gene approach, picking our candidates, and we were like the guy who lost his keys late at night, and after realizing that he wasn't going to get home without them, began to search. And where did he search? Well, here he was on the dark street. He searched in the one place where he could see, which was under the lamppost. And of course, that's not where the keys were. And that was exactly our problem. We were going on the hunches, which was like our lampposts, to try to find what were the genes involved in diabetes or cancer or heart disease. And frankly, we didn't know enough about these diseases to have very good hunches. And so for the most part, we, like the guy here in the picture, ended up empty-handed. Well, all that has changed, and it has changed dramatically. And most of this has happened in the last 2 years. And it's happened as a result of a project which followed right on the heels of the sequencing of the human genome, basically to try to look at that variable part of the DNA, that 0.5% or so that we differ in a systematic way across the world. Another international consortium, Japan again as a major player. In fact, this photograph is taken in Japan at one of our many meetings of the group. Again, I had the privilege of serving as the project manager of this enterprise, but it involved, as you can see here, a lot of different groups, and altogether close to 2,000 scientists working together to try to define, in very short order, this catalog of human variation. Once that succeeded, and it did, it then put into the hands of investigators who were trying to identify those genetic risk factors for diabetes and a long list of other diseases, the kind of catalog that they needed so that they didn't have to depend on hunches anymore. They could look at the whole genome. So no longer is this a candidate gene strategy. This is now what we call a genome-wide association approach, and now basically you're lighting up the whole street. You finally have the tools to search everywhere. And you know, the genome is a bounded set of information. It's big, for sure, but it's not infinitely big. It's 3 billion of these letters, and if you have a way of searching through it systematically, and you have people with the disease and people who don't, you should be able to find the differences. Is it working? Well, boy, is it working. The first success of this in 2005, here shown on a schematic of the human chromosomes, number 1 in the upper left and the X and the Y down in the lower right. The first success mapped a gene to chromosome 1 for age-related macular degeneration, the most common cause of blindness in the elderly, and came up with a gene called complement factor H, which was not on anybody's candidate gene list at all, a complete surprise, but which has shined a very nice new light on the reasons why people get this disease. And there it was. It just popped out of this analysis and has led to a complete revolution in our understanding of that disease and a whole host of new ideas about prevention and treatment. That was the first one. That was the only one in 2005. HapMap had not even quite finished its work, but again, putting all the data out there, people began to use it before we were done. In 2006, 3 more popped up. These are not easy studies to do. To be successful, you may need to study 1,000 people with the disease and 1,000 people who don't have the disease and scan across the whole genome in each of those people. So it's a lot of lab work and a lot of statistical analysis. But things began to happen. I'm not going to tell you what each of these diseases are, and I'm sure you can't read the fine print. It doesn't matter. Just watch the banners appear. Each banner here is a new discovery about a genetic risk factor in a disease about which we knew relatively little until this strategy came along. Now, 2007, things really started to take off. Just in the first quarter, the first 3 months, a whole bunch of other banners appearing, new disease findings on all these chromosomes. Second quarter, things going pretty wild and crazy here. Lots of diabetes discoveries, including some from my own laboratory during that 3-month period. The third quarter, continuing to add decorations here. The fourth quarter. Now we're into 2008. This is showing no signs of letting up. And here we are as of July 1st. And I guess it's about time for me to upgrade this, but I'm running out of real estate on the slide. I may have to figure out another way to display the results. Counting it up now, more than 200 of these discoveries. Remember we had 7 in 2002? Now we have over 200. Most of them arrived in the last 2 years. And each one of those shining a bright light into the reasons why a particular puzzling disease seems to strike some people and not others. Now, don't let me overstate this. Most of these risk factors are individually rather modest. If you have the risk spelling of one of these genes, your risk of the disease might go up by 20 or 30%. Generally, it doesn't go up by 4,000%. There are a few like that, but very few. But each one of them is telling you something about that gene is playing a role in that disease, and that tells you something about treatment that we could not have guessed at before, which is truly exciting. I should say we also have a project which is looking at genetic misspellings that are not inherited from generation to generation, but misspellings that happen during life and which cause cancer. And that is the Cancer Genome Atlas, being done jointly with the Cancer Institute. This project aims to look at tumors and to do in a very systematic, comprehensive way what's previously been done a bit like looking under the lamppost, and that is to scan the whole genome and find out in a particular cancer what is every little thing that has gone wrong. And if you look at the newspapers tomorrow morning, I think you will see a flurry of excited reports about what this project has done and which is being published tomorrow about brain tumors. A condition, of course, which is particularly frustrating and frightening in that this condition, when it does occur, is these days rarely cured. And as you know, Senator Kennedy, recently diagnosed with this, has brought that to people's attention who perhaps were not as aware of how this condition called glioblastoma multiforme can be so frightening. And in that very tumor, we now have a discovery of several new genes that are critically important and suggest new ideas about treatment that we could not have guessed at before. And that's the leading edge of what is going to be an outpouring of new information about cancer in the next few years, using the tools of genomics to take a comprehensive look at what has previously been sampled but not looked at in this kind of thorough way. So how is this going to play out in terms of its impact on medicine? I don't have time to go through many examples, but let me just point out the general ways in which these discoveries are going to find their way into the clinic, and some already have. One of those is, of course, the identification of these risk factors. That's what I was just talking about in terms of all those banners I showed you that have popped up on the diagram in the last couple of years. That is all basic science. What's the clinical implication? Well, one that comes along almost immediately is the opportunity to then use that to predict who's at risk for what disease, even while they're still healthy. And this would be a moment for me to ask you to think about whether you want to know. If I could give you the opportunity to find out your risk factors for a dozen common diseases over the next few decades at a reasonably accurate level, would that be information you'd like to have? Or would you just as soon go on without that and pretty much wait until something happens? I think a lot of people vary a bit in their answers on that. I think many of us look at it and say, is there something I could do about it? If you're going to tell me I've got a risk of Alzheimer's disease that's 5 times the normal, but there's nothing I can do, well then spare me. Maybe I don't want to know that. But if you're going to tell me I have a risk of Alzheimer's disease that's 5 times the normal, and here's what you can do to reduce that, then talk to me. Tell me about what I might be able to start doing today to try to change that outcome. Well, that would be a diagnostic attached to a preventive medicine strategy. And that really is the goal of a lot of what we're talking about here. And in some instances, we're already there. Here's a family with 2 individuals with colon cancer, 1 with uterine cancer. This is the kind of family for which we are now able to do rather precise predictions about who's at risk for cancer based upon DNA testing. And in fact, in this family, the 3 individuals in orange have in fact gone through that testing and are found to be at about a 60% chance of getting colon cancer during their lifetime on the basis of something they've inherited in a particular gene. Those individuals, however, are not just given this information and told to have a nice day. They're given the chance to begin colonoscopy at age 30, not age 50, but age 30, and to do so every year, not every 5 or 10 years. And in that circumstance, because colon cancer doesn't appear overnight, it goes through a long series of steps beginning with a benign polyp, these individuals can be assured that following that protocol has a very high likelihood of preventing an ultimate very bad outcome of metastatic disease from colon cancer. And in fact, one of these individuals has already had several polyps removed, which if not discovered at the age of 38, might have gone on to give cancer by the age of 50. So this is already standard of care today for individuals with family histories like this for this condition and a few others. Another area that is quite exciting, but for which the actual application is still not entirely broadly available, but it will become so, is called pharmacogenomics, which is a lot of syllables, but it's pretty simple, really. Think about it. When you get given a drug for a particular condition, sometimes it works pretty well. Sometimes you don't seem to get the benefit. Sometimes maybe you get a side effect. What's that all about? We're all different. Our response to drugs is different. So you give a bunch of patients with the same diagnosis the standard approach, and a lot of them will do well, but some will get no response or a toxic effect. We should be able to predict that ahead of time for a lot of drugs as we get smarter about what's in the genome. And already we're pretty close to doing this for the most commonly used drug to do blood thinning for people who have a blood clotting problem, a drug called warfarin. And there are a variety of other drugs for which this is being pursued, and a few for which it's actually been now approved as something that should happen. So don't be surprised if you go to your doc in the next 4 or 5 years, and before writing the prescription, the doc wants to do a DNA test to find out whether that's the right drug at the right dose for you, or whether there ought to be some change made. Ultimately, though, I think the main promise in the long term of understanding the genome is that it does allow us to identify the targets for therapy that are much closer to the action than what we currently know about. Frankly, as a physician, I can tell you most of the time when we treat somebody with an illness, we're not quite sure what we're doing. We're not quite sure why that drug works. Oftentimes the drug has been arrived at empirically, and we're not even quite sure how it's interacting with the human body to achieve a good outcome. Genomics allows you to strip away all of that confusion and go right to the heart of the matter and to pinpoint exactly what pathway is deranged in a particular disease and how you might treat that. And that gives you the chance then to develop rational drug therapies or gene therapies that are not empirical but are based upon a precise idea of where you need to affect the system. Perhaps the most compelling example of this comes from cancer, and it's the drug Gleevec for using now very effectively to treat an adult form of leukemia, chronic myeloid leukemia. Leukemia arises because of a genetic mistake in a blood cell in the bone marrow that results in the production of an abnormal protein. That protein is capable of taking a normal white cell and causing it to grow out of control, which is what leukemia is, and that's a blood smear with way too many white cells in it, here stained in purple, compared to the red cells, which are those faint pink ghosts you see in the background, which ought to be the majority of the cells. This protein that's produced in this type of leukemia has an active site, that little mouse bite there in the pink part of the leukemia protein. And if you could block that active site with some other small compound, some small organic chemical, then maybe, reasoned Brian Drucker of Oregon, you might be able to stop the bad consequences of that protein being present. And that's what the drug Gleevec does. It basically, in a lock and key fashion, fits into that ATP binding site. And the results of this have been nothing short of spectacular. The first trial of this in humans, the drug was given to 32 patients. They all had very advanced leukemia. They'd failed every other form of therapy. Most of them were not expected to live more than a couple of months. Of those 32, 31 of them had a complete and sustained remission, and many of them are at work today doing quite well, continuing to take the drug, but with no evidence of recurrence. That's the kind of outcome that one likes to see, and seeing it in this instance and in a few others gives us hope that this approach to rational drug design is going to have profound benefits once we have sufficient amounts of evidence to know how to do this and the research support to make it happen. This is complicated, this is expensive, this is high risk. You can't expect these kinds of promises to come true without very strong support for biomedical research in the coming years, but the promise has never been greater than it is today. So those are all, in a nutshell, some of the things that I'm excited about, and anybody working in this field is excited about in terms of the transforming capabilities of genomics for medicine. But what about these ethical, legal, and social implications, which people have also worried about, given our newfound abilities to manipulate DNA? One of the things I'm proudest about with the Human Genome Project was that these were identified at the very beginning as an important component of the project. The ethical, legal, and social implications, or ELSI component of the project. And I'm particularly glad to be here today talking about this because the very person who started the ELSI program in the Genome Institute is now here at Case, and you'll hear him in the panel after a bit, Eric Juengst. And Eric, when he came to NIH, charged to set this up, put in place a grants program that has encouraged work by dozens, in fact now hundreds of researchers from legal expertise, from social science, from theology, from a wide variety of other kinds of experts who have now become really capable scholars in this field and might otherwise not have been so. In fact, it's been estimated that the research support for ELSI in the genome arena is the largest amount of money that's been spent on bioethics research since the world began. We have really created, in this case, a circumstance where bioethics is not often the corner or something to think about later. It is part and parcel of this genomics revolution. And that, I think, has been a successful experiment, but it is an experiment and it's ongoing to see exactly whether we end up with better outcomes. All too often in the past, we judged, science has kind of gone forward, and then one day somebody woke up and went, oh my gosh, we should have thought about this ethical consequence. That's not the way we're doing it this time. The charge to the ELSI program is to think ahead, to be prepared for those issues that are going to vex us, and to make suggestions about how to plan policies to limit any kind of untoward consequences and to maximize the benefits. And that's what I think ELSI has quite successfully been doing, but needs to continue, obviously, as the science goes forward. I'll just mention a few of the questions that have been particularly prominently addressed by this program. Certainly anybody in the field of genetics cannot discount the fact that human genetics got off to a very bad start. And it got off to a start involving eugenics, and if you thought that was an excess conducted by the Nazis in Germany, think again. In fact, the foundations of eugenics were really laid in the United States particularly at the Cold Spring Harbor Eugenics Record Office by Charles Davenport and others. Eugenics being the science of the improvement of the human race by better breeding, which then led to involuntary sterilization and ultimately to the horrors of the Holocaust carried out to that extreme. We must not forget the lessons of the past, that this kind of misunderstanding and misapplication of genetics can have horrendous consequences. And as much as we would like to think that could never happen again, we have to keep looking at the lessons from that experience where scientists and physicians were in fact complicit in many of the actions that took place, and therefore cannot confidently say that this could not happen in the future. My own sense is that state-sponsored eugenics is rather unlikely to occur in any democratic society in the future. But the sort of homemade eugenics may be another matter. I'll come to that in a minute. What about our knowledge of human variation, which is exploding with projects like HapMap? How does that play into our understanding of what the meaning of race is? Well, what does race mean anyway? Evelyn Brooks Higginbotham has a wonderful statement that everybody talks about race as if they knew what they were saying, but if you ask somebody to define what they mean by race, they will arrive almost immediately at puzzlement. We really aren't quite sure, are we? Because there's so much baggage attached to this word. Yes, race is connected in some way with ancestral geographic origins, but there's a lot more than that. Race also carries with it culture and history, oftentimes difficult histories, by groups that have been discriminated against. And it carries other ideas about customs and diet. And so when you see, for instance, that a particular population is suffering a health disparity, that is to say, a particular disorder seems to be more frequent in that group than others, say diabetes, for instance, in Pima Indians, one should not rush to the conclusion that that means that it's on a genetic basis. It may have nothing to do with genetics. It may have to do with the environment and all the other components of that. And that's a very high priority issue right now for our LC program and for our research program, is to try to sort that out. One way to look at this is to consider that amongst the folks that you see here, their DNA is 99.5% identical, but that 0.5% means that each of them is facing potentially a different statistical risk of certain adult diseases by the group that they belong to. Now, if that's the case, if self-identified race or ethnicity is connected somehow to health or disease, what's the connection? And I would argue that you can think about this usefully in two different pathways. One, and perhaps the most prominent one in most health disparities, I'm going to argue, has nothing to do with DNA. It has to do with environmental exposures that are differentially distributed depending on which group you're part of. And that includes educational opportunities, that includes access to health care, that includes cultural practices, socioeconomic status, and even something like stress. Now, yes, it is also true, though, on the other side of the diagram, that self-identified race or ethnicity does correlate with ancestral geographic origins, that is something I can actually guess by looking at least some of you. If I took a DNA sample, which part of the world would your DNA sample most closely cluster with? But it wouldn't be very precise, and it certainly would be confounded by people who have ancestors from multiple different parts of the world, as many of us do. That in turn, that ancestral geographic origin does correlate a bit with genome variation, and it therefore is feasible to say that in some instances that connects to particular genes that are associated with disease risk. And that may in some instances, as we've recently discovered for prostate cancer in African Americans, contribute to a health disparity. But I would think that in most cases, when the dust all settles, what we'll learn is that health disparities are enormously complex, and there is some mix of environment and genes, and it'll be different in each instance, but we need to figure that out. And the other thing this diagram should remind us of is that if you want to treat people successfully in terms of prevention and treatment of disease, you shouldn't be using the thing at the top of the diagram to make your decisions. So race-based medicine is bad medicine. What you want to do is to get to the more proximal factors, specific environments for that individual, specific genetic variants for that individual, and then you may be able to do something much more useful. And the things at the top here are these muddy proxies that don't necessarily get you very far if what you're trying to achieve is better health. What about intellectual property? We've certainly been vexed by that one. I'm very proud of the fact that in 1996, when the international sequencing leaders got together for the first time as a group, including people from Japan and the UK and France and Germany and the US. China came along a little later. We had a discussion about that very issue, and this is a photograph of the whiteboard that was written on during that meeting, now some 12 years ago, in which the principles that we discussed adopting were placed. And you'll notice what it says, maybe that third bullet: aim to have all sequence freely available and in the public domain for both research and development in order to maximize its benefit to society. This past month, as I've been cleaning out my office at NIH, as I've moved away from directing the Institute and into a search for some new calling, I've gone through boxes and boxes and boxes of documents related to my 15 years there. And one of the folders I came across was the meeting folder from this very meeting. And I looked at my own handwritten notes from that meeting, about 12 pages, because we were at this for 2.5 days. And I came to this place, and I have exactly the same words written down. I'd copied them into my notes to be sure I had a record of them. But when I came to them, at the end of the discussion, I wrote rather cautiously, "I think we have agreement on this." I thought, my gosh, is that all we ever got was "I think we have agreement"? Well, I kept reading. And what I'd forgotten is we came back to that on the last day because people wanted to be really sure that we were all together on this. And we rediscussed those same points, and I think that's where the "and in the public domain" got added, just to be absolutely clear what we were talking about. And then I wrote in big capital letters, "unanimously agreed." Now that was a moment. That was a moment where a group of scientific leaders, without necessarily having the approval of all the authorities in their respective countries, but just doing the right thing, made a decision to absolutely change the ethics of data release in a profound way, not waiting for publication, not waiting for anything, releasing the data every 24 hours. And that attitude of immediate data release has now extended well beyond that decision 12 years ago into many other areas of biomedical research, I would say with great benefit to the public because of the way it accelerates the discoveries that need to happen next and levels the playing field. So anybody with a good idea has a chance to start working right away. Here's one that I think was identified right from the beginning as a particularly important one for attention. And in fact, I'll show you a timeline over the last 18 years of how this particular issue was struggled with as our LC experts and policy experts and scientists all got together and said, we got to do something. Because if we don't fix this, people are going to be afraid to find out about their own DNA because they all get discriminated against in the next breath by losing their health insurance or their jobs. And that is not just. You didn't get to pick your DNA, I bet. I didn't either. Should that be used to take away your access to health care or to a job for which you're otherwise well qualified? No, that is unjust. That is unsustainable. And frankly, if you think about it, it puts all of us at risk, not just some people. If you came here this afternoon thinking you were the perfect genetic specimen, I got really bad news for you. There are not any. And so if your genetic glitches, and you've got a few dozen of them, are going to be reason enough to lose your health coverage, you should worry about that. So we needed to do something about this. And this has been a lesson for me in just how complicated it is to take research and to turn that into policy recommendations and then to actually implement them, because our process doesn't immediately embrace an academic policy recommendation and say, oh, well, of course we should do that. There's a lot of other things at work here, and some of them are called special interests, and they don't go down lightly. So genetic discrimination was this object sort of pointed at all of us, and we needed to do something about it. So in 1990, the Americans with Disabilities Act was passed, and that had some potential of being helpful here, although not very. States began to take action about this in the 1990s. I'm not going to read each of the points on this series of slides because time is running short, but you can see there's a fair amount of interest. In 1995, the middle item there, Representative Louise Slaughter, Democrat of New York, introduced the first federal legislation about genetic discrimination, H.R. 2748. And Senator Mark Hatfield, my dear friend from Oregon, introduced a companion in the Senate. And we thought, okay, we're getting somewhere. We got legislation in the Congress. This is just going to happen, right? Well, it sat there. No hearings, no debate. That's what happens to most bills, by the way. In 1996, HIPAA, the Health Insurance Portability and Accountability Act, passed, and after a lot of behind-the-scenes effort by a lot of people, including people in my institute, and a lot of late-night discussions with members of the Congress, that bill did include genetic information on the list of things that can't be used to take away your health insurance if you're in a group plan. But it doesn't say anything about individual plans. And obviously, none of us know whether we're going to end up needing an individual plan at some future point. You don't know whether you're going to be self-employed 10 years from now. And so this wasn't all that reassuring to people who were fearing genetic discrimination, and cases began to appear where that was really happening. Time went on. Every odd year here, Representative Slaughter or Biggert would introduce the bill. You remember, Congress runs for 2 years, so on the odd years, you have a new Congress, introduce the bill, nothing happens. In 2000, President Clinton issued an executive order which basically provided genetic discrimination protection if you happen to be a federal employee, but that's all the president can do without an act of Congress, so that was somewhat limited. 2001, another odd-numbered year, bills introduced again, and in that same year, President Bush aligned himself with the proposition that something ought to be done here, which was encouraging. We have a president saying this needs to happen, people in both parties saying it needs to happen. It doesn't sound like this is a partisan issue at all, but it's so hard to get things going. Time is passing here. 2003, bills introduced, nothing happening. 2005, bills introduced again. This time the Senate passes the bill unanimously. S306, the president restates the support. I thought, finally, something's going to happen. The House didn't take it up, and so it died at the end of 2006. Well, happily, this story has a good ending. So in 2007, the bill, now called GINA, the Genetic Information Non-Discrimination Act, introduced in the Senate by Senator Snowe, in the House by Representative Slaughter, and on April 25th, which happens to be DNA Day, because that's the day Watson and Crick published their paper in 1953, the House passed this version 420-3. And we thought, the Senate's already passed this bill, right? 2 years ago? This is a snap. Well, one senator held this up for 1 year minus 1 day by objections that didn't seem to most of us to have a lot of merit, but were supported by some of the special interests that really didn't want to see this happen. But on April 24th, 2008, the Senate did take up the bill with a few modifications, passed it 95-0. Because of the modifications, it had to go back to the House to get considered again. And on May 1st of this year, GINA passed the House 414-1. You want to know who the one was? Ron Paul. I'll say no more. So then we were sure. We were there. We had a wonderful session. Senator Kennedy came over from the Senate for this moment of celebration. Here you also see Congresswoman Slaughter sitting next to Senator Kennedy, and Congresswoman Biggert sitting next to me, the two in the House who had really pushed this faithfully over all those years. It was only a few days later that Senator Kennedy was diagnosed with a brain tumor after having a seizure. Yet the people in the Senate who have pushed this forward, no one deserves more credit than Ted Kennedy. And yes, just 20 days later, the president signed the bill into law in a ceremony in the Oval Office that I got to be present for, making this now the law of the land. By the way, as he was signing this, I expected that somebody would get the pen, and I kind of thought that would be really nice to have after all these years of working on this with all of these various team members. So I went up afterwards and asked if I could have the pen. Well, it turns out they go to the presidential library. That's not allowed. Very frustrating outcome. But the president did send me a copy of the bill that he personally inscribed as a next best thing, which I'm very happy to have. So we now finally are in a circumstance in this country where one need not fear that genetic information about you will be used for discriminatory purposes in health insurance or in the workplace. Now, of course, that doesn't solve all of our problems either. What about genetic testing? Is it subject to sufficient oversight? Can you be sure if somebody's trying to get you to take a genetic test that the results are trustworthy? Does it actually tell you you're at risk for diabetes, or might that be wrong? And of course, that becomes particularly of interest right now because there are 3 companies out there marketing directly to the public, not through health professionals, but directly to you, the opportunity to find out your DNA sequence at about 1 million places in your genome, and to give you then a report card of what your risks of disease might be based on that information. The science behind these credible efforts is pretty good. The problem is, this is all very new stuff, and we don't at the present time have really good ideas about the interventions that you might want to consider if you're found to be at high risk. We could guess at them, but it would be nice if we actually knew for sure what would be beneficial. So is this premature? Are we jumping the gun here? And are we particularly jumping the gun by doing this in a fashion where no health professional is directly involved? It's a company and you having this exchange of information. A serious consideration a lot of people are wondering about, and again raises the question about whether there ought to be some kind of government oversight, which at the present time for this kind of testing, there's virtually none. Now, you don't want to squash a new industry that might be valuable, and many people are interested in this information. At the same time, are we doing a service to the public by letting this go on without some kind of oversight and intervention to be sure that you're able to identify circumstances where the information is not reliable? Along with that, if there were healthcare providers involved in these kinds of interchanges about the entire genome and risks of disease, would your doctor or your nurse be able to tell you what this information meant? Not necessarily. Most healthcare professionals have had no training in genetics. This has come along very quickly. They're also struggling to try to figure out how to make sense of this. An organization that I have had a hand in founding, and which is actually having its annual meeting today and tomorrow back in Bethesda, Maryland, is the National Coalition for Health Professional Education and Genetics, and this is their website if you want to look at it more. Basically, they're trying everything they can to put into the hands of health providers information that's well validated and objective about how this could influence the way they practice in their own medical practice, because obviously it's not something that you can necessarily read about in the textbooks that were written 10 years ago. And what about enhancements? I think a lot of people in the public are particularly worried about that. So yeah, fine, go ahead and cure cancer, that's good. But what are you going to do when it comes to trying to make the next generation a little smarter or a little bit more athletic? Are you tinkering? Are you playing God? What is going on here about enhancement? Most of the scenarios for enhancement are totally unrealistic. Folks, we will not be in a circumstance to modify the DNA that gets passed from parent to child in humans anytime in the foreseeable future. That would be unethical because we really would not know the consequences for generations to come, and that won't pass muster with any ethicists that I know. So the kind of tinkering we do with the genome is going to be of a rather limited sort, maybe in the liver of somebody who needs liver attention, or somebody who has cystic fibrosis. You might try to modify the DNA in their lungs to cure their disease. But those don't present major ethical challenges. When it comes to enhancement, though, I think the kinds of things people are worried about in terms of future generations often devolve down to that scene in the Gattaca movie where the couple is being offered the chance to basically pick the embryo that they could create that would have the best mix of features to make them feel like they had a high achiever for a child. And I understand the students over here watched that particular scene from Gattaca yesterday, and it is instructive, although it's pure Hollywood. Basically, the idea of PGD, or pre-implantation genetic diagnosis, which was initially developed to allow couples at very high risk for a disease like Tay-Sachs disease, is it allows you in an in vitro fertilization circumstance to do the in vitro fertilization, to take a single cell off of each of the multiple embryos that you have, to do a DNA test on that embryo, and to decide whether it's going to give rise to an affected child or not, and then you reimplant the ones that are testing normal, and don't reimplant the ones that would be affected. And while people vary in their views on the ethics of this, I think many people see that as acceptable for a early onset fatal disease like Tay-Sachs, are more troubled by the idea of using it for something like breast cancer as an adult risk, and are very troubled about the idea of using this for enhancement. But you know what? The enhancement scenarios aren't going to work anyway, so I think this is one we could sort of put to rest. The genes that are involved in something like intelligence or athletic ability are going to be multiple. The individual genes are going to have very small effects. You would have to have zillions of embryos to pick from in any given couple to be able to optimize for all of those in one embryo. The statistics won't get you there. And so this strategy in terms of enhancement is really doomed, or while it might be successfully marketed by some in vitro fertilization clinic who's trying to make money off of some yuppie couples, those yuppie couples will not be happy with the outcome because Junior, who was supposed to play first violin and get A+ in math, will be up in his room smoking pot and playing heavy metal music because they forgot about parenting part, which is probably a whole lot more important than the DNA. So don't worry too much that this is just about to burst forth on the scene. Finally, and I know I should have wrapped up a few minutes ago, and forgive me, I kind of got going here. A final question, though, that I think has to really be given very serious thought by all of us is in this enthusiasm, which we all have for the revolution we're in the midst of, and believe me, I have enthusiasm about that, are we beginning to get a little too carried away, and are we ascribing to DNA basically properties that it does not deserve? Are we thinking of ourselves increasingly as hardwired by our DNA? And in the process of that change of mindset, are we beginning to neglect other aspects of what it means to be human? Namely the experiences that we've all had interacting with the environment, interacting with our parents as children. And will this lead us, therefore, to pay less attention to the environment, less attention perhaps to education, and to downplay the importance of the human spirit and our need for God? That would be a profound misunderstanding of the science. Even when we have completely understood the human genome, we will not understand free will, nor will it have gone out of style. And nor will we understand, of course, why identical twins, when you get to know them, are actually very different people. They have the same genome after all. So that's telling you there's a lot more to being human than just what's written into those 3 billion letters. And as somebody who is both a scientist and a believer in God, I think we have to be very thoughtful about the ways in which in our discourse we are beginning to incorporate conversations about DNA as if it had some sort of significance it doesn't deserve. If you look at these two images, which I'm very fond of juxtaposing in just this way, it kind of makes you think a little bit about this. On the left, the rose window of York Minster Cathedral. On the right, a view of DNA, not from the side, which is the way we usually see it, but looking down the barrel, the long axis, giving this beautiful radial pattern that's at least somewhat reminiscent of that rose window. In my view, those who would argue that one of these worldviews has to win and the other has to lose are mistaken. And that's not a future that I think will be good for us or is in fact based upon truth. I think it is entirely compatible to both embrace science as the way to understand how the natural world works. It's a very reliable and powerful way to do that. And to recognize that spirituality is not necessarily approachable by science in the same way. And certainly the question of God's existence is not a science question, nor will it ever be. And so to have both a spiritual and a scientific worldview, and to have them exist in the same person at the same time without a firewall between them, is for me entirely comfortable. And it troubles me when I hear around us shrill voices arguing that one or the other of these has to go. I think if that particular perspective, those extreme voices, win out, we will all be very much impoverished as a consequence. And that is what I wrote about in this book called The Language of God, which puts forward from my perspective how it is that one can be a rigorous scientist who doesn't accept any conclusion about nature until I've seen your data and thought of all the reasons why it might be mistaken, but who also believes in a God who's the creator of the universe, who planned all of this in an incredibly elegant way to make it possible for complex organisms like ourselves to exist and to have conversations like this one. And may it always be that as we get deeper into the science, we don't lose track of the humanity. In the words of Albert Schweitzer, we must not allow our technology to exceed our humanity. Not now, not ever. Thank you all very much. So much. Oh, goodness. Thank you.
Dr. Collins will respond to 2 or 3 questions, and there are microphones at the front here. You can use them, and then we're going to transition. We won't take a break, but we're going to transition to the symposium and conversation where you'll have the opportunity to ask some more questions. Who wants to ask the first question?
Oh, good.
President Snyder was recently quoted as saying that she prefers to under-promise and over-deliver. I'd like to know whether you think the Genome Project has hewed to that philosophy.
That's a great question. It depends on whose promises you're referring to, I guess, and at what time they made them. I think at the time the Genome Project was reaching its conclusion, certainly in the flurry of attention around 2000 in that particular unveiling at the White House with the president, there were wild statements being made about how this was going to completely transform medicine overnight. None of us engaged in the project could see how that could possibly be correct, but I'm not sure we did enough to try to discourage that view, so perhaps by not strongly countering it, it was assumed that we shared that same perspective. I have actually, since about 2000, made a series of projections between 2000 and 2030 about how I thought this would play out, with many of the most dramatic implications for therapeutics not coming on board until 2015 or 2020. I would say now, some 8 years into my own timetable of those projections, it's about where I thought it would be. There's an old saw that says when you have a technological development of considerable significance, there's almost invariably an overestimate of its consequences in the short run and an underestimate in the long run. I think that may well turn out to be true here. Yeah, there was a lot of hype in the early part of this century about all of this. Some of it was used to raise a lot of money and to get a lot of stockholders to throw cash at organizations that had no products, and they didn't necessarily get their money back. But I think in the longer term, we are likely to be really surprised by the outcome in ways that we can't even stand here today and anticipate, and that might be quite revolutionary in ways that we have not had the vision yet to be able to interpret.
I'm a little more cautious perhaps than you are, and I think the Gleevec example is incredibly impressive, but I also think it's enormously unrepresentative, in that the genetic change involved is not one that would be discovered by the Genome Project and was known prior to the Genome Project.
Sure.
And in many cases, when genes are identified as being genes that are inherited are identified as being associated with disease, they offer really very little prospect of being a drug target. And the simplistic models that are generally held up as the way it's going to work for drug development often don't correspond to the reality. And a perfect example is sickle cell disease, where the currently useful therapies do not target the gene that was identified as the Mendelian locus of interest, but in fact, empirically, it turns out, target a different locus in the hemoglobin system, as you are no doubt aware. Sure. So I would like to propose that I think even though your time frame is perhaps appropriate of 30 or 40 years, that I think it's going to be the exception where genetic information leads to really effective therapies, whereas I think in diagnostics it's going to be important.
Certainly diagnostics will be much more straightforward, much more immediate. I'll give you 2 other examples, though, where I think the optimism that I've displayed is not entirely unwarranted, although you're right to express the skepticism that these have not yet been proven. One would be in the case of type 2 diabetes, where we've now discovered 16 new genes that were not previously identified in genetic analyses as playing a role in the disease. When you look at that list, most of them are surprises. Interestingly, 2 of them on the list turn out to be the targets for 2 very well-known classes of diabetes drugs. The thiazolidine diones, which are basically against this gene called PPAR gamma, and the sulfonylureas, which are against a gene called KCNJ11. Both of those turn up in the screen. If we didn't already know that those were incredibly valuable drug targets, we would have just discovered them. If that's the case with the 2 we know about, I'm betting that amongst the other 14, there are going to be some winners there as well. And in fact, there may be circumstances where it's not the gene itself that turned up, but it's the pathway that it points to, which may be more druggable than that particular gene. The other example, which is very recent and not yet published, but having just had lunch last week with Bob Beall and Melissa Ashlock of the CF Foundation, I am really excited to see the way in which this drug called VX770, which is completely derived from understanding what the cystic fibrosis gene's normal function is, appears to be having a profoundly beneficial effect on a subset of people with CF who have a particular class of mutations. And those are people who, after 14 days on this drug, have their sweat chlorides almost normalized and their lung function, as measured by FEV1, going up by 50%. Just an unbelievably good response in a 2-week period. Now, whether that will be sustained, I don't know, but it sure makes you start to believe that in this paradigm of gene discovery to therapeutics, it's not just Gleevec, and it doesn't have to take 40 years. There are circumstances that we can go faster and do better. But thanks for your question, and I really appreciate the skepticism. We need to all keep that in mind.
Whose responsibility is it that the techniques that you've been using in the past 5 years from computer science be used in the other sciences? That, I mean, biology and genetic epidemiology have been on the forefront of these techniques, but I don't see them in economics or the other sciences. Is it biology's responsibility to tell people what they're doing, or is it computer science? I mean, do you understand my question?
I'm not sure I understand the responsibility to tell people exactly what?
The methods that you're using, which are really sophisticated, complex computer science algorithms that aren't extending to the social sciences. I mean, is biology and genetic epidemiology supposed to play a leading role, or is it everyone else's responsibility to play catch-up?
So if I understand, you're sort of raising the question, are we making the most of cross-disciplinary opportunities to bring along other sciences that may not be in the midst or the center of this revolution? Yes and no. I think one thing the Genome Project has done is to encourage a lot of disciplines to talk to each other who didn't used to, and that's really wonderful to see. So you can't be involved in genomics without knowing computer science, because that's an awful lot of what we do. And engineering becomes critical, along with biology and medicine, and even chemistry and physics are in there too, and certainly automation and optics. Social science, I think, has been brought into the under the umbrella through this ELSI program, but I would not say, and I'd be curious what Eric Juengst would say about this, I would not say we've been completely successful in doing the melding of those disciplines in the way that maybe you're asking, have we achieved? Because you would like to have spin-offs of these various tools and technologies and algorithms into other areas where people haven't necessarily thought of them. Genome has been borrowing a lot of those algorithms from other places, and I think they have been borrowed from us, but we could always do better at that. One more question, yes.
Yes, sir. I'm very humbled to be speaking in your presence and a little bit nervous. But we had a question of a scientific and a technical variety, and I was wondering, on a more spiritual dimension, incorporating science, if you had a word or two to say to someone like Richard Dawkins, what would you say?
Richard Dawkins and I debated in front of the editors of Time magazine, and that got published as a cover story in Time about 2 years ago, which is still up on the web, so you can see some of that interaction in a more lengthy way than I will try to put upon you right now. Actually, Richard is a very motivated purveyor of the argument that evolution and other aspects of science mean that God is no longer relevant. And he's really quite angry about the whole thing. In particular, what he sees is the way that religion is being used in damaging ways, both within families and across the world in terms of terrorism and other excesses. I've recently had lunch with Richard. We've talked about these issues. I consider him an interesting intellect to have exchanges with, sort of jousting about these things. I think if there is a place that I would really hope Richard could move on a bit, it is to gain a better understanding of what it is that believers actually believe. Because in Dawkins' books, the way he caricatures believers doesn't represent anything that I recognize, and nor does it seem to fit the views of most mature believers. So he takes the tack of misrepresenting what belief is all about, and then finds it fairly easy to dismantle. I would rather he'd spend some time really immersing himself in what it is that causes mature believers to stick to their faith, despite all the arguments that they're just as familiar with as he is.
Thank you.
Thank you.
Seat over there. While the conversationalists, panelists in your program are coming up to the stage, I'd like to tell you about them and also what the rules of the next hour or so are. First of all, as you can tell from your program, and if you read the biographies in the middle of the program about them, you'll see what distinguished people they are. Why did I choose them? If you have any gripes, you can blame me. I chose Eric Juengst for reasons that you all probably appreciate. His long association with Dr. Collins and his international reputation as being a thoughtful ethicist. And I've had plenty of opportunity in the past, over the past 2 years, to verify that. I chose Georgia Wiesner because she is on the firing line, on the front lines of having to make decisions, clinical decisions with families, with individuals every day. She, in her job, which she's now stepped down from, but she's still very active in the Center for Clinical Genetics at University Hospital. And then, why Cynthia Beall? Cynthia is not in biomedicine. She's a world distinguished anthropologist. She's a member of the National Academy of Sciences, but she does not use the human genome for biomedicine, but she does use it for her research in anthropology. So this is largely unscripted. In fact, it's wholly unscripted from now on. I hope you can be participants in this unscripted activity to get things going. One, and I'm not quite sure which of our panelists is going to ask Dr. Collins a question. The panelists and I met about 3 or 4 weeks ago, and I can tell you we could have gone on all morning. So I think if we left it to them, they would go on all afternoon. After about 15 minutes or some period of time, I'm going to step back up here, and then we will begin the opportunity for you to ask questions, both of Dr. Collins and our conversationalists. So you're on.
I'm hoping that I get to be the first person to ask the question, and also just to congratulate you on the prize today. The breadth of work that you've done has been truly impressive, but the social aspects are really quite important. One of the things that we intersect in our careers is patient care. How do patients understand this? And genomics has created something that's now called personalized medicine.
Yes.
You put the little quotes on it. And you touched on some of the areas of where personalized medicine would go and could go. But what I was wondering is, as we do this, I actually have patients now coming in and saying, can't you just fix that gene? Can't you take that, turn it around, and fix the disorder in the family? So is that personalized medicine, or what would your vision be, and how can we help these people with their questions?
That's a great question, Georgia, and I am actually just starting to write a book about personalized medicine, so I've been thinking about this a little bit. I think what we're currently referring to, although there is a difference here in people's definitions, is the idea of trying to take the one-size-fits-all approach, which is often characterized the way we have handled things in medicine because it was the best we had, and trying to consider individual characteristics, particularly about DNA, to try to optimize the approach. And that means instead of telling everybody you should have a colonoscopy starting at age 50, figure out who's at the highest risk and start a little sooner, and who's at the lowest risk and maybe start a little later. And maybe instead of recommending PSA for all men when they reach that age, to figure out who at a genetic level is at risk of early onset invasive disease and maybe start sooner. And with some of the others say, you don't need this, it's just going to worry you because PSA is not that great a screening test, as we've recently heard recommended by august panels. Try to do that sort of optimization. Likewise with drug treatment, instead of just the standard so many milligrams for this person taken this many times a day. Could you do better if you understood all of the P450 enzymes in that person and made the appropriate adjustment? And certainly when it comes to therapeutics, and cancer is going to lead the way here, if you have a brain tumor and you know that the particular genetic glitches in that tumor are the following 6, and that they aren't the same 6 as somebody else with that tumor, and you have an array of drugs that have an array of targets, you ought to be able to personalize that approach more effectively than the current, okay, you have this tumor, this is the cocktail you get. Now, how realistic is that? In some ways, it's here. In other ways, it's not, and it's not clear how long it'll take. But moving in that direction, I think, is what people are excited about. There's all kinds of challenges, not just with the science, but with the sort of health policy questions. Who's going to pay for this? Since we don't pay for prevention anyway, we have a healthcare system that doesn't seem to care very much about prevention. They'll take care of you once you get to the ICU, but wouldn't it be nice if we kind of kept a few people out of there? We have a big issue there in terms of management. Now, don't get me wrong. I'm not saying this is all that brand new. I mean, when you get a blood transfusion, it better darn well be blood typed for you. That's personalizing. We've been doing that since Landsteiner told us how to. If you're having a kidney transplant, it had better be matched to you, or it's going to get rejected pretty quick. So some of this personalizing has been standard of care, but a lot of it is more recently arriving on the scene. And it's hard to argue with the principle, but it's also, I think, challenging to say exactly how to implement it with what we know now.
Well, and I think one of the areas that patients have trouble with is what you were kind of talking about before, which is the deterministic idea of some genes, and that they may, in fact, have more strength in terms of the component of the disorder, and that it's not that they should be exercising, watching their diet, doing those sorts of things that they might be able to do for their health as well. So I think people try to find that one marker that will explain the risk that they can have that pill taken.
And I think some of the way in which these new discoveries are being portrayed encourages that view. If you've seen the Discover magazine that's currently on the newsstand, it's all about direct-to-consumer DNA testing as a way to find out what your future is. And it seems to imply, at least until you dig deeply into the article, that this is a lot more precise than it is.
One of the images that often goes along with those stories is the crystal ball. Yeah. The geneticist as literally fortune teller. And that's a pretty powerful, magical form of prediction. And it seems to me, I think you would agree, that probably we need another image for...
A weather predictor. Isn't that yours, Eric?
That's right.
Go ahead, hit them with it. It's a good one.
The TV weather map as the image for what genetic testing can provide to you. We know that TV weather maps are probabilistic. Our experience of the weather is going to change with the local environment. They don't try to tell us what to do, particularly to prepare for the weather. So it seems to me that's much closer to the kind of risk information we're going to get.
And it's getting better over time, too. So we hope the same will be true with genetic prediction, right?
So I have another question, though, and that's to pick up on the theme, again, of the virtues of personalized medicine, because it does seem to me that the virtues there lie in the direction of its potential for prevention. It's really what's cool about it is
I wouldn't say it's all of it, but it's a big part of it.
In front of diseases, right? So prevention itself has some interesting possible ethical and social implications. And I wonder what you would think about either of these kinds of cascades of developments. In preventive medicine, we always talk about levels of prevention: primary, secondary, and tertiary prevention. Primary prevention is stopping people from getting infected by germs in the first place by cleaning up the sewers or the food supply. And in preventive medicine, that's always the ultimate goal, to keep people from getting sick in the first place by stopping the spread of the causes of disease. Won't the same logic apply in genetics, pushing us back further and further to say, well, we do have that pre-implantation diagnosis technique Francis was talking about. Why not use that to do primary genetic prevention and avoid the birth of people with these risk factors in the first place? So
now we're getting also into the sort of eugenics territory.
It's the back door into that.
Yeah, as I probably not very accurately went through, this would really only be barely imaginable in a single gene condition, because otherwise you have not enough embryos to sort out what you'd like to sort out. But say a single gene condition. Even there, if you're talking about a recessive condition, you might be able, if you systematically applied PGD to every pregnancy, to avoid a birth of a child with Tay-Sachs disease or some other rare recessive. But you wouldn't change the carrier frequency very much in the population, because most of the Tay-Sachs mutations don't reside in affected kids. They reside in parents who are carriers who are entirely normal. And even with that kind of systematic effort, if you go through the math, it takes very, very many generations before you have much of an effect on the gene carrier frequency. In a dominant, maybe you could see something like this having an effect in a shorter term, say Huntington's disease, where in fact, if we currently think there are very few new mutations, so they shouldn't be popping up de novo. If you could try to identify all of the people who carry that and systematically screen every pregnancy, and of course that's being done by some couples because they want to do it for their own reasons, then maybe you could change that gene frequency. But you see how limited it would be? It would be a very, very tiny slice of the medical ills that affect human beings that could be manipulated in this way.
But could you imagine a family saying, for example, we've had such a problem with breast cancer in our family that I just as soon not even pass on the risk factor, much less the disease.
And this is a hot topic right now, as you know, in terms of ethical discussion. Is it appropriate to use PGD for an adult onset condition for which medical advances may be happening? And are you really doing something to prevent an illness that may turn out not to be that important by the time that person grows up and faces the real risk of disease? There's just been, as you know, in the UK, a debate about this in terms of PGD applications. In the US, where PGD is not really watched over at all, there's virtually no regulation or oversight on in vitro fertilization, which is where this happens. I am sure there are lots of examples where exactly your scenario has been pursued by a highly motivated family. Whether that's a good thing or not, or justifiable, or fits with ethical norms is somewhat debatable. Of course, this is the classic example where you have a collision between ethical principles. You have the principle that a couple is supposed to have primary ability to decide what they want to do about childbearing. And then you have the principle that society is invested in fairness and in valuing individuals and in valuing diversity and not sort of expecting everybody to be perfect. The handicapped community, of course, is very concerned about this creeping attitude that the only acceptable human being is one that has absolutely no evidence of any disability. And I think we have to listen to them very strongly in that regard.
And we can ask, would you want to make that kind of life and death decision on the basis of a TV weather map level of information?
Well, exactly, because for things like breast cancer, you're not at the level of precision that maybe is being portrayed in some situations.
So the other cascade of issues that is a little more futuristic, I think, it goes in the direction of enhancement. Because I can imagine that many forms of improvements in the human design will come under the banner of prevention. We want to be able to upgrade our immune systems to seek out and destroy cancer before it even starts. Does that worry you, this idea that prevention is a gateway to redesigning the human?
It's certainly a possibility. In fact, when you think about it, we are already there, aren't we? We all vaccinate our children.
Yes.
So, in fact, if you don't, you're considered to be a bad parent. And after all, what is that? That is a biological enhancement.
Right.
And you are actually rearranging DNA in your T cells and B cells when you give a vaccination. That's how it works. And we're comfortable with that. Yeah. And yet it is exactly what you're saying. It's sort of starting down the road in the name of prevention that actually could carry a label of enhancement as well. Enhancement is a funny one because a lot of people will say, well, I'm against enhancement, but then they're in favor of piano lessons. And they may not be too bothered about fingernail polish or hair coloring, or even plastic surgery for cosmetic purposes. So when you're saying enhancement, what is it that really bothers you? And it's often messing with DNA, seems to take you to another level of concern. And of course, one of the cases I've tried to make is we need to make a distinction there, a spectrum between something that is truly beneficial and admirable, like the enhancement from vaccination, and something that we would all recoil from, right. Which would be enhancement to try to help an athlete be better at doping, basically, on a genetic basis, which is feasible in some instances. And then we also need to put all of those opportunities on a scale of possible today, maybe possible in 10 years, and never possible, because some of them are really completely out there scientifically. And then try to focus on the ones where there's a real possibility of changing the outcome. If there are things that are potentially regrettable and short-term, let's think about those. And let's not worry about the designer babies that are never going to happen in the Hollywood fashion.
Well, I'd like to move from the personalized medicine to global health, and to think about, to hear your thoughts on how genomics can be exported and applied to improve health globally, where there's so many countries where the health conditions are so different, as well as human biology varies.
Sure. No, and I really appreciate your raising that, because I think all of us need to lift our eyes from where we often have them focused right around our own communities and think about the rest of the world. One of the things that I have found most rewarding is to go and volunteer in a hospital in West Africa and see what it is like for individuals who don't have all the resources that we do, and what kind of medical care is feasible in that setting. And it's life-changing to have that experience. I think we all wish that every new advance that came out of research could immediately be exportable to every possible place in the world where it could be valuable. Realistically, that's not the way our world is currently organized. What I learned working in this mission hospital in Nigeria was, though, that it is a good thing that advances were happening in Europe and in the US and in Japan, because they did find their way there. It took longer than it should have, but it did happen. And so the medicine one could practice was maybe 20 years out of date, but it was a lot better than it would have been otherwise. I think we need to make a really serious effort, though, not to just hope that happens, but to catalyze it. And this is an area of profound current interest on my part. I think, for instance, there is a whole host of diseases that you do not see in this country, beginning with malaria, but going on through some of the parasitic diseases like leishmaniasis and trypanosomiasis and schistosomiasis, for which genomic approaches to understanding the pathogen and how it interacts with the host could give us new ideas about vaccines and drugs that would be vastly more exciting than anything that's currently there. And in fact, that's one of the efforts that I helped start at NIH before leaving, was an effort to specifically target those neglected diseases using the tools of genomics. Obviously, everything you do there, you have to keep in mind the economics, and that if you develop some wonderful intervention, but it's not affordable in the environment that needs it, then you haven't done anything. And it certainly is wonderful that there's more interest here from places like the Gates Foundation, but we need a lot more if we're really going to get serious. And just as a bit of an editorial, I hope our country can take this more seriously in the future. We have a tradition now, we have an image in the world of being a really good soldier for the whole world to basically be afraid of, but could we actually change our view a bit and think of ourselves instead as the scientist and the doctor to the world? We would both be doing a lot more benevolent things and potentially improve our image and basically be living up to the promise and the responsibility of a country as wonderful as ours. And I think we've slipped a long way from that responsibility, and my hope is maybe in the next few years we'll get that dynamic turned around.
Let me intervene temporarily here. We may have the opportunity to come back and give our panelists other opportunities to ask questions. Did the John Hay School students have to leave for their bus? Is there anybody from John Hay that's still here that was going to ask a question? That's my, we had originally intended to have one of their students ask a question, but I guess if you're here, raise your hand. Well, I guess you're off the hook. So does anybody who's been listening to this, or even if you haven't been listening, have a question for Dr. Collins or any of the panelists? Dr. Foley, I see you.
Can you unleash your speculative self and tell us what genetic practice will look like 10 years from now?
Would you please repeat Dr. Foley's question?
So Dr. Foley is asking, could I unleash my genetic self? And I'll ask Georgia to unleash herself too and say what genetic practice of medicine would look like in 10 years. 10 years from now, it will be possible to sequence any of our genomes for $1,000 or less. And I think there will be great interest in just doing that once and for all and putting that information into the medical record, because then you won't have to arrange a rapid DNA test at some future time. When you need it, it'll just be there, and you'll simply click with your mouse and find out the information that's needed, hopefully well protected for privacy purposes. So I think that will be the case. The human genome will be part of your medical record for most of us within 10 years. How will that information be used? Again, I think the ability to make predictions about future risk are going to get better. Frankly, they're still pretty blurry right now. That crystal ball is very cloudy, or the weather map is not so precise. But a lot of the heritability that's associated with common diseases hasn't been discovered yet, and we know it's there from family studies, and it will get discovered, so that it will be possible 10 years from now to be able to make much more quantitative statements about an individual's risk for disease. And I would hope that that would lead to conversations between the provider and the individual about what they might do in terms of diet and lifestyle and medical surveillance to focus on keeping healthy in an individual, personalized sort of way. I think that's entirely realistic in 10 years. I think in 10 years, there will be at least a dozen drugs for which the evidence is strong enough to know DNA results before you write the prescription, that will become standard of care. I think we'll get there for warfarin before too long. There will be others. And so that won't be an unusual circumstance when somebody's being treated for a disease, is that drug dose or even drug choice is being individualized based on what's known about their genome. I hope we will know a lot more about the environment as well, and we haven't talked much about that, and we should have, in terms of how the environmental factors interact with the genetic factors, so that we can begin also to do more subtle things to encourage people to change their environment if they happen to be at high risk, and we'll know what to say about that. In terms of therapeutics, in 10 years, I think we'll have a lot of new and powerful drugs for cancer that'll come out of the current revolution in understanding cancer. We'll begin to see some of the new drugs for common diseases like diabetes and Alzheimer's, whose targets are just now turning up, but that's a long process, so that'll be still very much in the evolutionary steps of getting to where you want it to be. I would think, if we have any sense, we will have changed our healthcare system to allow focus on prevention instead of ignoring that. And I would think by then healthcare providers will have to be literate about genomics, or they will have a very hard time feeling as if they are providing the kind of care that all healthcare providers want to provide. That's a snapshot. Georgia.
Well, I think this is a time to be very, very optimistic about how genome sciences will change and continue to change medical practice. I was just thinking this 10-year time frame. My current students that are in their first year of the curriculum at the medical school, which is the new WR2 curriculum, what we are teaching students is to actually use and integrate genetic concepts along the way of their whole medical career. So genetics is one of the basis biologies that they need to understand. So I'm thinking of this class that will be seeing patients and taking care of patients. And if the way in which we're teaching students and the way we think the medicine will be practiced will be along the basis of understanding those pathways that might be affected, understanding a little bit more of the diseases, and understanding the phenotypes that come from those specific genetic changes, so that we can do that forward sort of thinking. What Dr. Collins didn't tell you is that you actually can get your genome sequenced now if you have $350,000. And wish to know. But I think the big issue is interpretation. How are we going to interpret what we are finding? What does that actually mean in terms of health and disease? And how do we translate that into prevention? So I'm optimistic, and I think it's the next few years there'll be so much growth and understanding along the way.
Can I unleash myself?
Unleash. Okay. Go ahead.
One of the things I think is going to happen in 10 years is the result of not human genome sequencing, but using the genome tools to sequence other organisms. We talked about malaria. Here, I don't know if Jim Kazura is here, but I'll blow his horn here at Case. They're sequencing both the mosquito and the amoeba that cause malaria in order to find out their secrets. Meanwhile, at the Genome Institute, they've started a big project to sequence all the bacteria that live in and on our bodies. We think about those as germs, but actually they're quite a vital part of our normal physiology. And I think probiotic medicines based on the results of that research are going to come out.
I do. Good point. We have a question here. Could you come to the microphone? Yes.
I'm wondering what you regard as the more significant research or work being done in the relationship between genetics and the environment.
So this is a critical area that needs more attention than it's had, and I think there's an effort now called the Genes and Environment Initiative, which is in its third year at NIH. In my view, a lot of the problem is we have not had great tools for assessing environmental exposures. We've come a long way in measuring the genetic circumstance by all these technologies that read out DNA sequence. The environment is much harder. You have an almost infinite variety of compounds in the air, in the water, in the food that may influence our health, many of which we don't even know about. And coming up with methods to accurately measure those is a challenge. You also have the problem that an environmental exposure today may not cause a problem until 10 years from now when the exposure is long gone. And how do you figure out what happened when you weren't there at that critical moment? There is a big effort to improve the technologies with a lot of money going into that, and particularly using real-time measures, including cell phones, to basically use the cell phone networks to be able to transmit information about people who are going through their regular life experiences, as opposed to filling out questionnaires about what you think you ate or what kind of air you thought you breathed. Let's actually measure it, and that will help. In my view, I'll make a little plug here. I will tell you there's one project that I very much hoped to be able to initiate while I was at NIH, and I have failed. And that it would be a project which would basically enroll 500,000 to 1 million people across the country from multiple different geographic areas. Race and ethnicity would be proportionately representative of the country, so would socioeconomic status and age and gender, and to for those volunteers who would basically be volunteering for a national study of health, learn everything you possibly could about them in terms of their environmental exposures using these new technologies, in terms of their genetics, in terms of their health problems, and have that go forward over the course of 10 years or longer, we would finally have sufficient power to understand how genes and environment interact for common diseases with a study of that size. It would be expensive. It would cost roughly $400 million a year. That's a lot of money at a time where research has been taking a serious hit for the last 5 years, with basically research losing its buying power each year to the point now where only 1 in 5 people sending a grant to the NIH has a chance of getting funded. And there's just no money to do something really bold like this. But every year that goes by that we haven't started this study, we are losing the chance of learning from it. And I hope that we might turn the corner soon and see the value of research in general and this kind of project in particular to be able to begin to build that information, because that's what we're going to want to know. We're not just going to want to know who's at risk. We're going to want to know what was the environmental factor that played a role in whether they got sick or not, and let's see if we can fix that. We're not going to change your genomes, but we could change the environment if we knew what to do. Right now, we're kind of in the dark.
I see at least 3 people, and first Dr. Singham, then Dr. Whitehouse, and then the young lady here, in that sequence. I think we are gradually moving from the benign questions into the ones that might be a little tougher, if I know Dr. Singham and Dr. Whitehouse anyway.
I'd like to thank you, Dr. Collins, for fighting to keep all this information in the public domain. Mm-hmm. And I'm wondering, are there still dangers that of people who would try to make information proprietary in order to make private profit at the expense of the public good? Are there some dangers that we should be aware of and be ready to combat?
Well, there's always that tension, and don't get me wrong, I think there are kinds of data about the genome that are highly appropriate to be claimed in intellectual property claims. After all, why do we have patenting and why do we have secrecy in some circumstances? In the good way, it's because it allows a discoverer of something to have time or a protected monopoly to be able to develop a product that the public needs. And we would not have most of the therapeutic advances that we count on for treating disease if it weren't for the patent system to enable that kind of investment to get made. What my argument would be is you got to think really carefully about which side of the line you're on before you apply that kind of business model, and that basic fundamental information about the human genome or about variation in the genome or even about how that variation correlates with disease risk for most of us is on the side of the line that says this ought to be public, there should not be secrecy, there should be immediate release, and there shouldn't be patents. Not everybody agrees, and I think a particular area of tension is going to be this area of diagnostics. So for instance, if I've discovered a genetic variation that predicts a high risk of heart disease, you could make a case that if I patent that and license it exclusively to one laboratory, they're going to get a lot of business and they're going to make a lot of money. I don't think that's good for the public. I think it's much better if there is some competition for people offering that test, which drives the cost down, drives the quality up, but it may not be what the university tech transfer officer says when they think they've got a chance here to finally make good on the president's commission to that tech transfer officer. You're supposed to be making money. And there is this tension that's always present there. So I think the bar has been shifting a little bit in various directions. Frankly, the 1990s were a very bad time for this. We had this gold rush where people were claiming snippets of DNA and filing patents and getting patents on them, even though they had no idea what those snippets did. I don't think people are trying to do so much of that anymore, mostly because it's all now in the public domain, and partly because even the patent office has shifted their view about what's appropriate. But we could shift back again if we aren't careful. And I just think that's a part of the many arenas of public discourse at a time like this, on an afternoon like this, that we have to continue to talk about. And it can't just be the scientists who are deciding where does that boundary lie between patentable and non-patentable. That's got to be a really big public discussion.
Dr. Whitehouse.
Thank you for creating this space to have these important conversations and for your contributions. I've been also fortunate to have funding from your former institute to study apolipoprotein E4 and its relationship to what we call Alzheimer's. Yes. And it's also related to risk for heart disease. It may also be protective for macular degeneration. So the whole issue of pleiotropy of genes makes personalized medicine considerably complicated. And we've also been looking for other Alzheimer's disease genes, spending a lot of money and not finding any with significant risk. So my concern about personalized medicine, it is complicated. And also, in the time that the physician talks to the patient about their genome stored on the personal computer, will the physician have enough time to talk to that patient about their spirituality? Something I would say is important to personalized medicine as well. And finally, my real question is...
You're on a roll, keep going.
There are a lot of ethicists and medical physicians who are focusing on genetics. The elevation of Bethesda, Maryland is about 300 feet, and I know that because I wondered how threatened it would be, NIH, by global warming. And do you think that perhaps we need to get doctors focused on ecology and environmental health in balance to focus on genetics?
Whoa, okay. Answer, yes. And actually, that's one of the opportunities I guess I'm now seeking a bit myself now that I've stepped away from the Genome Institute, is to really think more broadly about all of the challenges that face our planet at a time of many of those beginning to emerge as pretty serious. And I would certainly agree with you that climate change has not only the consequence of economic problems, and obviously problems about properties that are potentially going to be underwater, but also about medical implications. As climate change occurs, the globe is warming, then natural habitats for pathogens are shifting. Malaria is being found in elevations where it didn't used to be. And all of those changes are going to require a serious medical and public health interventions that have only barely begun to be contemplated. And, you know, sometimes you sit and think about all of the things that we have to try to face at once between our health challenges, the climate change, our energy difficulties, the wars that seem to pop up, and we never seem to quite know what to do with them. And you can get pretty overwhelmed and pretty pessimistic. But I guess, like Churchill, I have faith that somehow that people will figure out how to address the problems in a thoughtful way, and maybe not in an efficient way. We'll muddle through somehow, but we're not going to do so without the best and brightest minds of this generation and the one that comes after it, turning their attention, not just to genetics, although it's a pretty cool thing to work on and it ought to be on the list, but we got lots of other problems that are at least as important for attention. I appreciate your making that point.
Right here.
Okay, my question's a little bit off topic from what we've been discussing today, but we all know the medical benefits of sequencing the human genome. But what are your opinions on the use of it through other sciences like evolutionary biology, more specifically using sequencing of genomes of other organisms in finding common ancestry or common descent?
Well, it's clearly happening. So we have now draft or complete sequences on more than 30 vertebrates. You can feed those sequences into a computer without telling the computer which organism the sequence came from and say, how are these related? And the computer will generate a perfectly beautiful phylogenetic tree that matches almost precisely the evidence from the fossil record and the evidence from anatomic features that people have used in the past. The consistency of those trees is a very compelling argument for descent from a common ancestor. And when you look at the DNA details, it becomes even more compelling, where you look at things like pseudogenes, genes that have lost their function, which in some other related species still have worked. It's very hard to imagine that that's not related in terms of descent from a common ancestor, and in one species the gene was no longer needed and so it acquired some mutations, but it still left itself there as a DNA fossil in the genome. I think anybody who looks seriously at comparative genomics can really no longer defend the idea that common ancestry is not proven. It is incontrovertibly clear from the data. Now, the one thing that I think people could put up, and rightly so, is that the early stages are a little unclear in the sense of what was going on in that microbial soup before there were any eukaryotes. Because it's clear there was so much horizontal gene transfer capabilities in those early stages that the idea of figuring out what was the common ancestor or what was even a species at that point is really quite murky, and that's exciting to think about. But I don't think in any way it challenges the Darwinian view of how life forms are related to each other. Others want to comment on that?
That sounds good to me. I would ask, for example, when you showed that photo of a lot of children in school and saying that they shared 99.5% of their genes, that's a number that is often thrown out as the difference between chimps and humans. And I'm wondering, well, it depends on whose numbers you're reading.
Right, we've revised a bit lately for complicated reasons.
Okay. Well, then, what do you think we've learned that is very distinctive about humans from comparative genomics?
First of all, I do think it's fair to say that it's not as easy as it used to be to say how similar are humans to each other. If you lined up a stretch of DNA from me and from you in a place where we could align the two closely together, the difference would be about 0.1%, 1 out of 1,000. But then there's this category of copy number variance, where there may be a stretch of DNA that's tens of thousands of base pairs long, and I've got 3 copies of that and you've got 4. When you add those up, because it's a big stretch, that adds up to this other 0.4%. So now we've sort of revised our number from saying we're 99.9% the same, now we're 99.5. If you apply that same dual standard to us versus chimps, if you're just looking at the alignable part, it's 98.8%. If you throw in those copy number variants, it's less than 98%. So we're still not getting close to each other. It's not as if we're ultimately going to realize, oh my gosh, we are one species. That's not going to happen. What makes humans unique? Well, you know, there's zillions of differences here. Even though those are small percentages, the denominator of 3 billion is plenty big. And it's very hard to look at it and say exactly which of those matter. Now, an interesting view of that is coming from sequencing Neanderthal DNA, because here we have an opportunity to look at a dead end in Homo development, Homo neanderthalensis, where DNA has been retrieved from those bones from individuals who died 30,000 years ago, and the sequence is starting to emerge, which is pretty amazing to contemplate. And you can then look and say, okay, here's the chimp sequence, here's human and Neanderthal, which are much closer together, and you begin to draw conclusions then about what made humans particularly interesting. If you find a place where Neanderthals and chimps are the same, but humans are different, that means that happened in the last 500,000 years, and we know a lot of interesting things happened in terms of human capabilities in those 500,000 years. So that's going to narrow down the number of possibly really important changes, but it's still going to be a very big pile of opportunities to sift through, and we don't have great ways of actually assessing which of those matter because we aren't going to be doing experiments on mutating those in our own species. At least I sure hope not.
We're going to have 2 more questions on web. There's one here, and there's one over there. So let's do one in front of me first.
Georgia wanted to add something.
Oh, I'm sorry. Georgia, go ahead.
And that is that we've been talking a lot about coding DNA, that is DNA that actually make proteins and the proteins that are doing the work. We now know through many, many other scientists that there's great tracts of DNA for which we didn't understand their function, and that's now somewhat emerging. So there's going to be a lot more comparative analysis that needs to be done that will say this is human, this is not, within those non-coding regions. Yeah. So there's more to come.
Thank you. My name is Rhonda Williams. I'm an associate professor in the history department here. And thank you for your very thought-provoking talk and the engaging discussion so far of the panelists. I have a question, kind of a hypothetical, but I think a very practical kind of question. It seems to me that it's possible for us to act ethically while conducting genomic research, considering advances in private therapy, and also kind of considering the social implications of doing this kind of research in health disparities. And yet, health disparities can still exist. So I guess my question to you then would be, how do you move from quote-unquote ethics to social justice and equity, or have them march side by side, right? Because it seems like you can have one and not the other, or they could work against each other.
Yeah,
right? So
that's a great question. I'd be curious to see what Eric would say about this as well. It seems to me one of the reasons I told you the story about the Genetic Information Non-Discrimination Act in the detail I did was because of exactly this issue. That ethics research told us early on that on the basis of well-tried and accepted principles, genetic discrimination is something we should not tolerate. So it became a social justice question for which we had research evidence about the appropriate pathway. And in a certain way, genetic discrimination fits in the same category as civil rights. It's about discrimination on the basis of something you don't get to choose. It has a lot to do with your inheritance. And yet it took those 12 years for the handoff from the research conclusions and the argument about social justice to actually result in some implementation, in this case, legislation. The same will happen with health disparities. There's a lot of lip service that's paid by lots of people to how health disparities ought to be our #1 priority in changing the way in which medical care is delivered and successfully received. But of course, our healthcare system doesn't speak to that at all. When we have 46 million people who have no health insurance, and people who are in the health disparity populations are overrepresented in that group, then even with all of the brilliant insights that may come out of understanding the causes of those health disparities, be they genetic or environmental, what are we going to be doing about it if we have a healthcare system that remains utterly broken? So you can't really get there without fixing that problem, which is a political one that scientists like me can advocate for, but it's going to take a complete uprising of the public saying we're not going to stand for this anymore to really solve that. And I guess I'd like to think that's possible, but we don't seem to be that close to it right now.
Before Eric puts his 2 cents worth in, we're going to have one more question from the audience, and then we'll wrap it up. So just in case you're worried about those things. Eric, do you want to respond?
Yeah, sure. That's a good question, Rhonda. And I think, you know, in the biomedical world, the buzzword of the year is translation. We need to translate venture science into clinical benefits, into real medicines. Well, the same phenomenon applies to so-called ELSI research, ethical, legal, and social implications research. It's the need to translate what comes out of the academy, out of our social science studies, out of our philosophical analyses into political reform. And that's not an easy process. Look at the civil rights movement and how long it took from the moral perception, the ethical argument that something was unfair in this country to the passage of civil reform. Actually, genomic discrimination has been addressed exponentially quicker. If you think about it in those terms, it seems like a very long road for Francis. That's because he was in it and deserves a lot of the credit for getting GINA passed. But in the larger scale of things, those wheels have already begun turning faster than one would expect from if you're thinking about a major civil rights legislation.
Last question.
My name is Cheryl Scaccieri, and I'm from the Cleveland Clinic. I'm a genetic counselor. So there are so many things that we could talk about for hours here that have been discussed, and Dr. Collins, you've given everyone in the room a great foundation on genetics and then also the ethical issues that are being debated. One of the things that I wanted to focus a little bit more on was the eugenics movement and our history that in which really science was misinterpreted, biology was kind of twisted, I think, and also people who were trusted, physicians and the scientists, kind of were a big part of why the eugenics movement happened here and why there was involuntary sterilization of disabled individuals. But you talked also about, you know, conflicting ethics principles, and you also, well, I guess what I'm looking for is a way that people can become informed and contribute and participate in these discussions. You spoke before about vaccinations and how you're really manipulating people's DNA. Well, let me tell you, my pediatrician never said it like that to me. And so would the average person think twice if it was presented as, oh, there's this, this fancy thing happening in your child's T cells in the DNA. So I guess what I want to know from you is, part of it is the spin that we give these things as geneticists, as ethicists, and we're giving people some tools to evaluate, but we're not reporting it necessarily. We all have our agendas, and I want to make sure too that there's a place that maybe you can speak to this whole audience about where we can participate in some of these discussions.
So that's a great question, Cheryl, in terms of how do we live up to our responsibilities to actually explain what we're doing without shading it in a way that assures an outcome that we're hoping will be the outcome. I think that does happen from time to time. I think scientists have a great role to play in these social debates about what's right and what's not right about applications of science, but frankly, I think scientists will always be in a situation of having a conflict of interest. We like to do science, and we don't like people telling us, no, you can't do that experiment unless they've got a really good reason. And so whenever it comes to one of these tough ethical issues, scientists ought to be in the room, because otherwise sometimes the debate goes way off course and they're debating something that's not scientifically feasible instead of the real issue. But scientists alone making the decisions just can't be the right way to get there. And I think we have been experimenting with various ways of including many different perspectives in those debates. The genetic discrimination debates were like that. Lots of advocates got involved. The professional societies got involved in terms of caregivers. Policymakers got involved. Scientists were there saying, here's what we can do today, here's what we think we can do in 5 years, but they weren't making the decision. It's a pretty good model. But in other areas, I'm not sure we've gotten quite that level of engagement. And clearly, I imagine that's one of the things that is thought about in a place like this, and with an Inamori Center and with Eric's Center of Excellence in ELSI research. How do you really significantly involve multiple voices before you already have tracked something down the road towards the outcome that some subset is already counting on? And you really give it the chance to play out. I've learned a lot from the Native American community in this regard. Native American community is intensely skeptical of genetics. They have reasons for not being very trusting of what the government or the white man is trying to put over on them the next time, and they refuse to be brushed into anything. And they want to have a chance to talk about it and think about it, and no, don't tell me we have to do this today. And that has been, at times, intensely frustrating. But it has also been a really good learning experience about the responsibilities that scientists have to explain themselves and to really put everything on the table and not just the sort of nice wrapped package that you might otherwise think you're going to be able to put forward and not have too much debate. Debate's good. Sometimes it goes on longer than you want it to, but oh boy, compared to the alternative. Eric, what do you think? You must think about this in terms of setting up such opportunities for conversation. How are we doing on that?
Well, one of the things that's happened in the last couple of years from the NIH Roadmap Initiative have been the restructuring of clinical research in major academic institutions. Goes under the acronym of the CTSA program. I'm not even quite sure what that stands for. But
Clinical and Translational Science Awards.
Clinical and Translational Science Awards.
I'm from the government. I'm here to help with acronyms.
Yeah, that's right. But a significant piece of that is a community engagement program, because the NIH, I think, recognized that these major medical centers like UH and the Cleveland Clinic are located in communities, and we need to be involving the folks around us in the kinds of scientific planning and that we do. So that's happening in Cleveland, and we're excited about taking advantage of it through my center to talk about genetics issues. And then specifically, one of our projects on the drawing boards is a discussion of genomics and health disparities and what in the world it might mean to look for clues to health disparities in our genes. On the surface, at least, a pretty counterintuitive proposition. So,
before we finish, I want to make sure that we have been fair to DNA. We've been talking about DNA, I think, that's in our nucleus or nuclei, but there's another home for DNA, and I'm going to ask you, Dr. Beall, whether we have given short shrift to mitochondrial DNA and whether anything we might consider about mitochondrial DNA is relevant to the discussion today, and try to be this is just to bring it to our attention because we do need to finish.
Oh, well, that was a tough one out of left field. You did include the mitochondria, right, as you were sequencing the human genome?
It is definitely part of it, and it's part of these genome-wide association studies as well, looking to see if there are variations there that might be playing a role in disease.
Certainly the mitochondria have been used as Dr. Collins mentioned, to trace people's genetic ancestry. And recently there have been claims that if we test your mitochondrial DNA and some nuclear DNA, we can tell within 120 miles where your ancestors came from. So anthropologists, in fact, use mitochondrial DNA a lot. It's also cheaper.
Well, I want to thank our panelists and Dr. Collins and all of you for participating. We are finished.